Okay. Hi, I'm Jeff. I broke the one slide rule I have to because I like this cartoon. So
What's the
I'm
slide?
a lot of
One
people.
slide rule.
Yeah.
Oh,
A lot of people study interactions sort of between species or cell types or whatnot, but I'm kind of more interested in interactions within sort of one population, one species, sort of interest-specific ecology. And I have sort of a little cartoon. Yeah, I mentioned the assistant professor John. So these are cancer cells, but you can think of these as yeast or bacteria or viruses or whatever. But it's sort of the traditional view of resistance that we like to think about, where sort of and even before some sort of diagnosis happens, just through random mutation, you get a resistant cell. We apply some sort of drug, and eventually we get this sort of mostly resistant population at the end. And then what do we do to study it? We probably send it over to Vaughn's company to get sequenced, and we do a bunch of phenotyping, and we study the hell out of it in increasingly sophisticated ways. But something that's sort of a little sneaky is that when this resistant cell first emerges in the population, it's sort of one resistant cell in an otherwise ancestor or sensitive population. And none of the phenotyping that we actually do is typically done in that environment. We do it in this homogeneous resistant strain environment. And so sort of the question is, does that matter? And so we've done a lot of sort of characterizing of this question. Ishan was talking about doing it with these cancer cells where we did sort of frequency dependent growth rates of the two different populations. And we showed that they have dramatically different growth rates depending on what frequency they are in the population. So we see in cancer that this sort of question, this sort of environment that we're characterizing them in is quite important, and we can't really ignore it. So if we are interested in this evolutionary process, we probably need to do more co-culture experiments. And then now I'm sort of working on, we showed it in cancer, so I'm trying to show it in different systems. So I'm working in some yeast data. So if you can go next slide. So there's this beautiful paper that came out in 2015 that used barcode lineage tracking to do yeast evolution. So they evolved 500,000 barcode lineages in a limited glucose environment. They found about 25,000 different beneficial mutations, and they sampled about 4,500 of them independently. And they did an assay, and using this data set, I was able to recreate a frequency dependent fitness for each of these 4,500 mutants and plotted them on this graph on the right. And the point here is I'm just showing two of these points. Basically, how fit are they when they're essentially in monoculture, and how fit are they when they're very rare in the population? And if you're in the lower right-hand triangle here, which is where every single one of the 4,500 mutants is, you're more fit when you're rare than when you're common. So you have this frequency dependence, and it's not small. I mean, these are 50%, 80%, sometimes twice as fit, growing twice as fast when they are rare than when they're common. And again, it's every single mutant. So now we've seen it in cancer, we're seeing it abundantly in yeast, and then a recent preprint looking at some of the LTEE lines in bacteria show that there's an abundance of this frequency dependent growth rates going on there. So I think we've just sort of stumbled on something that we should have been looking at for a very long time that has massive consequences in population genetics that we've sort of been ignoring. And yeah, this is sort of what I'm going to measure when I start at Maryland in the summer. And sort of these are my first experiments, is building around this kind of stuff in yeast and bacteria.
the problem is PopGen's hard even with just intrinsic growth.
Yeah,
and then you put an off diagonal into things, and like
things get complicated.
things get complicated quick. Like single intrinsic growths, like the diagonal existing is hard, and then if you have rows the same, it's still hard. It's harder, but still hard. And you go off diagonal different, which is what you're. It's just friggin' a lot.
Yeah,
especially if it's more than two by two.
Yeah, exactly.
This is like lots by lots.
Right. And we like to call these sort of positive frequency dependent effects because the ancestor is often conferring some sort of advantage to the resistant, but in classic PopGen, this is negative frequency dependent selection. So a more evolution based training would understand it through that lens.
Jeff, awesome. I can just say that we've done a sort of, I think the right measure is shit ton of evolution experiments over the years, collecting a range of mutants, and they're almost inevitably negative frequency dependent. So,
Yeah.
you know this, but I'm adding to your pile. I'm
Excellent.
really curious about the like that data in the lower right from the Levy and Sandeep's collection, right?
Yeah.
Where did that data come from in that way? Did you
So
collect
the
that?
no, so Grant Fisher. So okay, so Sasha came up with like a fitness assay protocol where they
Yep.
isolate a bunch of these and then do a fitness assay. Uh,
Yep.
and then Grant Kinsler did sort of a second step mutation of these lines where they took some of the most common ones
Oh
and
yeah, that's right. Yeah.
in that experiment, they did this fitness assay version again. And in the fitness assay, they start with 95% ancestor, 5%,
Yep.
and they culture it for sort of like 5 dilutions. And
Yep.
by the end of the experiment, the resistant strains are effectively 100% of the population. And so you actually get frequency dependent, you basically get 5 different measures of growth rate over 5 different segments. And so you can extract a frequency dependent growth rate, but they don't do that. So
Yep. I'm
well,
so basically these data are in their PLOS biology paper.
exactly, yeah.
Except extracted differently.
But yeah, they don't extract the frequency dependence at all. So I've just like taken their data and what they published and cast it into my hypothesis,
I
basically.
love it. Well done.
Yeah, and then we got basically we have data from I think 6 different experiments now in yeast and one in bacteria, and they all show this phenomenon.
So isn't it weird, by the way, then that like a lot of those mutants are in RAS PKA?
Yeah,
And isn't
So
it weird? It's weird, right? So obviously this is something that you guys know better than we do about because it's pretty relevant for cancer adaptation too.
yeah. It's
That they're
well,
negative frequency dependent.
right. Well, what's interesting is yeah, in the Kinsler assay, there's like 7 different pathways that these mutations pop up in.
Yeah.
FPK pathway, but they all end up in this regime. Like they're all negative frequency dependent. So once I saw that they were so prevalent in all these different pathways, I sort of foregone the try and figure out a single mechanism and just
Sure.
at the systematic level. But that's the obvious next step is classifying different mechanisms. The I've done a little bit of modeling that shows that you can get some of this behavior if you're competing for a single resource in a serial dilution experiment. So in this case, they're competing for glucose. And the idea is that when they're rare, they get significantly more doublings off than when they're common because they're not
Yep.
resource. So if you're a really fast grower, you get more generations when you're rare than when you're common. So maybe that's sort of this common mechanism in this case, but that doesn't seem to be the mechanism in the cancer case. So it just seems like basically the pop gen and the simulation work I've done shows that if this phenomenon exists, it will always be the most prevalent mutants. And it's inevitable that this is the phenotype that we'll measure is something that grows faster when it's rare than when it's common. And so the question is, how common is the phenotype? Because if it's common, it's like the only thing we'll ever see. And if it's rare, then we'll see it only when it pops up.
Are you saying the top left of the triangle does exist, but it's just self-selecting to not be observed?
The top left is hard to exist because you only get your fitness benefit once you've taken over, and you need to first take over. And so the takeover part of it is heavily preferentiated by selection.
So it could be popping up, but just like so
Yeah.
transiently that it's never.
Exactly.
Yeah.